All right, well, like I said, I'm Madeline. Thanks for having me here today, and I'm from the National Genomics Center, which is located here in town, and I'm going to tell you a little bit about the Aquatic eDNAtlas project and how that can be a useful tool. So, starting off, as probably everybody in this room knows, there's a ton of challenges to invasive species management, and this figure is demonstrating how an invasion can typically occur. As time progresses, the invasion costs and the invasion area can increase. So the most effective management is often happening in the lower left-hand corner of that figure. However, to operate in that area, you need things like early detections across the entire potentially invaded range, and that often means really large-scale monitoring. Both of those things are really hard to do, and as we've been talking about, environmental DNA, or eDNA, is one of the things that has helped to fill that need. And it can do that because it is effective at fine-scale sampling over large spatial and temporal ranges. And it is effective in that way because pretty much anybody can go out and collect a sample. We have pictured here kind of the process. The plastic cup there in the water has a filter in it and it's attached to a pump. So as you're pulling water across that filter, any DNA that's in the system from organisms can be potentially collected onto that filter. You would then simply place that filter in some sort of storage material at the NGC. We use desiccant, and you would send that back to a lab for analyses. At the National Genomics Center, we use sensitive and specific qPCR assays that are either built or optimized in-house. And like was also just mentioned, because these assays are sensitive and specific, they can be really good at detecting species that are rare on the landscape. So this also includes the leading edges of invasions. This is a figure from Wilcox et al. 2016 that demonstrates the sensitivity of eDNA, or potential sensitivity. On the y-axis, you have the probability of not detecting a fish, and on the x-axis, different fish densities. The eDNA is in the black line. As you can see, it's really good at detecting fish, even at low densities. Because of all of these attributes of environmental DNA, this is something the National Genomics Center has specialized in over the last 5 or so years. The National Genomics Center is part of the Research and Development branch of the National Forest Service and is located here in town, like we said. It's special in that it brings together a really great group of hardworking and smart scientists, and some of those people are pictured here. And they really uphold a high scientific standard and have a long history of doing so. Because of that, over the years, we've had the opportunity to work with a lot of different collaborators. And one thing collaborators benefit from working with the eDNA program specifically is that we would help out from the very beginning to the very end of the process. So initial planning stages all the way to interpreting results. Another thing that the NGC can do, because of the diverse array of scientists as well as the diverse array of collaborators we get to work with, is operate at a really large scale. So that includes things like database management as well as spatial monitoring. And those are both things that will come into play for this eDNAtlas project. But before I get to that, I'm going to tell you a little bit about another project that showcases what the eDNA program can do, as well as became the beginning of the eDNAtlas project. So this project in particular was concerned with a native species, bull trout, which anybody in the Northwest is probably familiar with. But like I said, this project is kind of what became the beginnings of the eDNAtlas project, so it gives some really nice context. This started out, the bull trout inventory project, with the Boise Spatial Stream Group creating a predictive model to look at the probability of juvenile bull trout occupancy throughout the western United States. After that model was built, they also produced a pre-designated sampling grid with associated metadata. And after that grid was in place, lots of collaborators participated in going out and sampling at all these different locations, and then sending those samples back to the NGC for further analysis. This resulted in what you see on the left-hand side of your screen, right-hand side of your screen. The green dots are denoting samples where brook trout were absent, and the blue dots are denoting where samples where bull trout, were detected. During this large-scale project process, we realized that there's the potential for overlapping sampling effort in eDNA, as well as untapped potential for large-scale collaborative science, because each sample that is collected can be analyzed for multiple different species. So we decided we wanted to make all the information about these versatile samples publicly available. So using this framework of a pre-designated sampling grid with associated metadata, the eDNAtlas was born. The eDNAtlas is a website. It would look like on the right side of your screen if you were to bring it up on your computer. And it is an open source ArcGIS portal to the NGC's eDNA analyses. And as I've alluded to, this was made possible by a truly collaborative effort, both with scientists at the NGC and in the Boise Spatial Stream Group, as well as from the huge number of collaborators who already have samples represented on the atlas. Currently, there are approximately 100 different collaborators with samples on the atlas, and this consists of over 14,000 different samples and about 6,700 analyses. All of these numbers are expected to increase soon, with the next update to the atlas should be occurring in 1 to 2 months. It's also important to note that that huge number of analyses is across a suite of 40 to 50 different species that we've worked with different collaborators on. It's also important to note that we require express permission from all collaborators before posting their data on the atlas, and that all data undergoes a stringent QA/QC process before going on the atlas. And this is to ensure a high quality of data for anything you might find on the atlas. In addition to this website that's showing you the eDNA results, we also have websites that just have those pre-designated sampling grids. So if you were to go on this website, you could see all those pre-designated points, whether or not they have been collected at, and all their associated metadata. So that includes things like the gradient of the stream, average summer temperature, and upstream catchment area. Additionally, each point in these grids is linked to the National Hydrography Dataset. So if the information or metadata you're interested in isn't directly available on the website, you could find that same point in the National Hydrography Dataset and learn even more about it. So if you're interested in planning an eDNA project, this could be an excellent place to start. As a side note, if you are planning a project, we have all the equipment you could need for sampling at the NGC, which you can borrow. As far as navigating the websites, there is a user guide available online, so you can download this and it will help you navigate the tools on your own. But I'll take the next couple minutes to show a couple notable features of the atlas to familiarize you with it and get you thinking about how you might be able to use the atlas. To demonstrate these different pieces of the atlas, I'll walk you through looking at examples from a project we collaborated on the University of Washington with. This project was concerned with determining the distribution of invasive smallmouth bass in the Northwest. So what we have on the bottom left of your screen is zoomed in of the left-hand side of the website when you open it. That is a list of all the different species we have results for or will soon have results for. So once again, that's on the order of 40 to 50 different species. If I was interested in looking for the results from this particular project, because it's concerned with smallmouth bass, I would check smallmouth bass there on the list, and that would result in all of the data associated with any smallmouth bass sample populating the map. If I wanted to go through that with a more fine-tooth comb, there is an additional filter feature available on the atlas, which is shown there. I would once again choose smallmouth bass, and then because I know this project was published with the University of Washington, I'd use this secondary drop-down bar, select agency, and then University of Washington, and that would result in this on the website, which is all the samples only from that project populating the map. Another significant feature of the atlas is that you can download data onto your personal computer. An easy way to do this is to use the attribute table, which is going to appear on the bottom of the website screen. And then in that attribute table, clicking your options and export to CSV. There are a number of ways to populate that attribute table with your data of interest. One way is using this filter tool I just talked about and populating the map with your data, and then hitting the filter by extent button here. and that will cause all of the data displayed on the map to populate the attribute table for easy export. If you wanted to take a step back or decided you were interested in a different species, you could go back to the left-hand side of the website and select down here any species you want, and then click View an Attribute Table, and all of the data from that species would then end up in your attribute table for easy export. Or if you wanted to zoom in even farther in a particular project, you could zoom in on your map to, say, a particular drainage and use this highlight tool on the map here to highlight your data of interest, and then once it is highlighted, it would appear in your attribute table once again for easy export. So these tools within the atlas that I've just shown you, as well as the atlas in general, allows you to query the archives of samples that are at the National Genomics Center. And these samples represent a biodiversity archive for aquatic systems in the western United States over the last 4 to 5 years. And because each of these samples represents the biodiversity in the system at the time of collection, and because they can be stored for a really long time, you have access through the atlas to a huge number of historic biodiversity timestamps. Additionally, another use of the atlas, like I've talked about, is you can view where and when all the other samples have been collected. So there's this potential for you to increase your sampling effort or area without actually going out and collecting any more samples yourself. So, for example, if you're interested in monitoring all the way around Flathead Lake, which is just north of here, but you only have the time or the funds to collect in a small portion of that area, you could go onto the atlas, zoom in on Flathead Lake, and see that there's a huge number of samples collected all around the lake. You could then, through us, contact those collaborators and potentially test those samples for your species of interest. Of course, to do that, you would need to have an assay built for your species of interest. What I've displayed here is a subset of assays we have for invasive and nuisance species that are particularly relevant here in the Northwest. But like I've been saying, we have a huge number of assays for many things beyond invasive or nuisance species as well. I want to kind of finish off with an example of how the NGC archives has been used in combination with this brook trout assay to just give you an idea of what we can do. So this project was published in 2016 by Wilcox et al., and they used those archive samples to look at the effect of invasive brook trout on native bull trout here in Montana. One thing they observed were brook trout excluding bull trout at high abundances. Figure on the right-hand side of the screen is showing this. On the x-axis, there's different mean summer temperatures and mean summer discharges. And on the y-axis, you have the probability of bull trout being present. Each line with a different color is showing a different relative brook trout abundances. The higher relative abundances are lower down on your x-axis in the purple and in the red, and as you can see, those are resulting in lower probabilities of bull trout being present. So the new data that allowed this group to observe this was collected at a fraction of the time and cost as it would have taken to actually go out and get a new sample. Instead, they actually just used these archived samples. Additionally, this allowed for a temporally paired data set, which they would not have had otherwise if they'd had to go and collect a new sample. The last point I'd like to make is that we are developing a pipeline to include samples analyzed at other agencies, because the goal is to have as wide a net as possible cast for all the data we can get on the eDNAtlas. This does pose a number of challenges, as different agencies might use different protocols, so there would need to be additional QA/QC measures to ensure a high quality of data for the eDNAtlas. We are also considering including metabarcoding data going forward, because there is a lot of people out there doing that. However, we're not currently set up to include metabarcoding data on the atlas. Very last, I'd just like to encourage you to think about how you can use the atlas going forward, both for invasive species as well as any other species you might be interested in, as this is a very versatile tool. As I've alluded to, this has been a really collaborative effort, and there's a number of scientists both at the NGC and in the Boise Spatial Stream group who have contributed. So some of those people are pictured here, and much credit is due to them. If you are interested in learning more about the atlas or working with the NGC, this is some contact information. My information is listed first, followed by Tommy Franklin, our program leader, who is also in attendance today. Dan Isaac is an excellent person to direct technical questions about the website. And if you have questions about the NGC as a whole, the contact information for Michael Schwartz, our director, is listed as well. And the last thing up there is the website address for the eDNA Atlas. Thank you all very much for listening. Are there any questions? Yeah. Oh, yes. So differences between what we're doing at the NGC is we're using qPCR assays. So typically we're just looking for one species or one very specific group of taxon with that, and that's what we're detecting. Whereas metabarcoding is you're kind of sequencing everything you find in your sample and then figuring out who those sequences belong to. So it's it's a less specific approach, and it results in many more hits for many more different species. So it's very different than just, did we find this piece of DNA or did we not? And so that's one of the challenges for presenting it on this website that was initially set up for like, yes, we saw it or we didn't.